Hello, everyone. This is Parinaz Iskandari from Electrical and Computer Engineering Department, Western Michigan University. On behalf of my co-authors, I am presenting our research work titled Flexible Microplasma Discharge Device for Treating Burn Wound Injuries Against Fungal Infections. Here is the outline of my presentation. First, I'm going to present the introduction by explaining about sterilization and why is it important for burn wound infection related complexities. And I will explain about current sterilization methods and what is plasma and microplasma treatment as a sterilization method. And in experimental section, first I'm going to present the fabrication process and screen printing process and how to prepare pathogen for these experiments and the whole treatment process. And at the end, I will present my results and conclusion and future work as well. Sterilization describes a process that eliminates all microbial life forms, including bacteria, viruses, and fungi. Microbial life forms that causes infections are called pathogens, and many of these pathogens may cause irreparable problems, especially when they enter through wounded skin. Therefore, sterilization of wound is considered to be the first and important step in treating the wounds. The National Center for Injuries Prevention and Control in the US reported about 5,000 deaths each year from burn-related complexities, and 20 to 25% of common microorganisms causing burn wound infections are from fungi species. And Candida albicans is an opportunistic fungal pathogen which is resistant to chemical antifungals. So proposing an efficient approach for elimination of Candida albicans is so important. Current sterilization methods include steam and dry heat, chemical and plasma-based sterilization methods. A steam and dry heat-based sterilization involves special chambers and requires heat chambers for sterilization, and hence are not portable and bulky. Chemical sterilizations utilize toxic chemicals, and if not disposed properly, can cause environmental pollution. Similarly, traditional plasma-based sterilization is not only bulky and not portable, but also utilizes chemicals such as hydrogen peroxide and ethylene oxide. To alleviate the use of chemicals and chambers, microplasma-based sterilization methods utilizing ambient air as the sterilizing agent can be used. The question is, what is plasma? Plasma technology is based on a simple physical principle. Matter changes its state when energy is supplied to it. Solids become liquid and liquids become gases. If even more energy is supplied to gas, it is ionized and goes into the energy-rich plasma state, the fourth state of matter. Plasma contains ions, electrons, excited atoms, and ultraviolet or UV radiation, free radicals, and chemically reactive neutral particles. Microplasma-based devices can be further categorized depending on the type of microplasma discharge. The microplasma jet utilizes a gas discharge through a nozzle containing the electrode that provides the high electric voltage required to form the microplasma jet. Surface microplasma is the type of plasma discharge where the plasma is generated on top of the functional electrode. Microplasma can be operated in a controlled chamber as well as in open air. However, the use of a controlled chamber adds to the cost of the system and makes the system very bulky. So discharging ambient air is more economical and cost effective. The advantages of the mycoplasma-based sterilization in ambient air is its use of ambient air as the sterilizing agent. This reduces costs significantly as there is no need for special chemical agents or chamber required. This improves its portability and are of smaller dimensions than traditional sterilization devices. The use of lower operating voltage is also a significant advantage, also reducing operational costs and bulkiness of the system. The mycoplasma discharge devices also has comparable sterilization efficacy to traditional sterilization methods. The advantages mentioned makes mycoplasma-based sterilization using ambient air as one of the ideal methods for use in surface treatment and wound disinfection. In this work, a mycoplasma discharge device was designed and fabricated. And here you see the materials which were used to fabricate this device. PET from DuPont Teijin films and the thickness of 50 micrometer was used as substrate, and silver ink AG800 from Kayaku Advanced Materials were used as the electrodes, and double-sided Kapton tape from Bertec in the thickness of 125 micrometer was used as encapsulation. In the bottom picture, you could see the fabrication process. First, PET was cleaned with IPA, and in the next step, silver ink was printed on PET substrate in a honeycomb design as top electrode. And in the next step, silver ink was printed on PET substrate in a circular design as bottom electrode. And finally, two films of PET attached to both sides of double-sided Kapton tape. In this work, to do a printing of silver ink on PET substrate, a screen printer from semi-automatic screen printer was used due to its less waste, more volume production, lightweight, and easy to fabricate. In the bottom left picture, you could see the screen printing design into honeycomb design and circular design as top and bottom electrodes respectively. And here is the screen specifications. The screen size was 12 by 12 inches, mesh type stainless steel ultrafine diameter. Mesh count was 325, mesh angle was 22.5 degree, and emulsion thickness was 15 microns, and image size was 6 by 6 inches. And in the bottom right picture, you see the fabricated mycoplasma discharge device in this work. To investigate the antifungal efficacy of the mycoplasma discharge device fabricated in this work, first we need to prepare Candida albicans. Here, a culture of Candida albicans was prepared in yeast and mold or wine broth and incubated at 37 degrees Celsius for 24 hours. Then a serial dilution was performed from 10 to power minus 1 to 10 to power minus 8 to determine the colony forming units or CFU on Wyam agar in Petri dishes. The purpose of this serial dilution was to achieve countable colonies on agar plates, and the criteria for being countable is that the number of colonies must be in the range of 20 to 200. And in this experiment, the forced dilution was considered to be the control, and all treatments were done with this dilution. In this experiment, the control had 72 colonies, when it was plated on the agar plate. When the treatment is done, the whole samples were put in the incubator for 24 hours in 37 degrees Celsius. In this slide, I'm going to present the treatment process. Here you see the experimental setup in which DC power supply is connected to DC to AC inverter, and it was connected to microplasma discharge device. The red wire is connected to top electrode and the black wire is connected to ground electrode. And when DC power supply is applied, microplasma discharge device turns on. And when it turns on, we could place the wire agar in which Candida albicans was spread on top of the device to be treated. Here you see the results of treatment of Candida albicans. In this experiment, control has 72 colonies and treatment time is fixed to 60 seconds. And by increasing input voltage from 6 to 10 volts, the number of colonies decreased significantly from 72 to 0 colonies. Actually, in this experiment, by applying 10 volts in 60 seconds, 100% sterilization was achieved. This experiment replicated and standard deviation was calculated in each step. And in this slide, you could see the results of treatment, but this time input voltage is fixed to 6 volts and treatment time is was increased from 60 seconds to 120 seconds. And as you see in this picture, by starting from 60-second treatment time and increasing the treatment time to 120 seconds, the number of colonies decreased significantly to 4 colonies. Actually, by increasing the treatment time from 1 minute to 2 minutes and a fixed input voltage of 6 volts, 95% sterilization was achieved. And this experiment was replicated, and again, standard deviation was calculated in each step. The fabricated microplasma discharge device in this work is going to apply to treating burn wound injuries, so its surface temperature is an important character. And here you see the image which was taken by FLIR thermal camera from the entity in on mode. And in the right graph, you could see the surface temperature of this device by increasing voltage from 6 to 10 volts. And as you see in 6 volts, the surface temperature is 22.36 degrees Celsius, which is safe for wound sites. As a conclusion of this work, I would mention that here in this work, a flexible microplasma discharge device was developed for eradication of Candida albicans using ambient air as a sterilization agent. The antifungal efficacy of the microplasma discharge device to eradicate Candida albicans was investigated by testing the effect of varying parameters, including input voltage and treatment times. An efficacy of 100% sterilization was achieved by applying 10 volts input voltage for 60 seconds. And an efficacy of 95% sterilization was achieved by applying 6 volts input voltage for 120 seconds. And surface temperatures varying from 22.36 degrees Celsius to 67.52 degrees Celsius were observed as the input voltage was increased from 6 to 10 volts respectively. Future research will focus on measuring the efficacy of the fabricated device in eliminating other pathogenic microorganisms from ESCAPE family pathogens. Thank you.